Hello, future doctors. Kaise ho aap sab? Welcome to today's high-yield revision explainer. So today we are tackling a truly massive topic for MBBS and NEET PG exams, which is cytoskeletal filaments and cellular junctions. Let's just dive right into this. We're going to break down these concepts so clearly that you won't just memorize them. Aapko class mein hi sab yaad ho jayega. Grab your notes and let's get started.
Okay, तो आज का roadmap जल्दी से देखते हैं. We'll start with a quick intro to the cytoskeleton, then move straight into microtubules, microfilaments, and intermediate filaments, along with our high-yield clinical correlations. Finally, we'll wrap up with cell-cell adhesion, tight junctions, and gap junctions. Let's begin.
All right, Section 1, Intro to Cytoskeleton. The etymology here is pretty simple. Cyto means cell, and skeleton means bone. Just like our own bones, this is the cell's internal support system. It gives the cell its physical strength and is completely responsible for cellular movement. Now, if we look at them based on size, microtubules are the big ones. They handle massive transport and heavy support. Microfilaments are the tiny ones, primarily taking care of basic cell motility. And sitting right there in the middle, we have intermediate filaments. The absolutely crucial exam pro you need to know here is that intermediate filaments are the most abundant filaments in the entire cell. A direct question aata hai exams mein, so definitely lock this into your memory.
Moving right along to Section 2, microtubules and clinical syndromes. Get your mental highlighters ready, students, क्योंकि यहाँ से clinical questions बहुत ज़्यादा आते हैं. This is exactly where foundational basic physiology perfectly meets pathology. We are looking at a neuron here, specifically focusing on axonal transport. Think of microtubules as these long railway tracks running all the way down the axon. Important materials need to move forward from the cell body, down to the synapse, and then waste or return signals need to move in reverse, back up to the cell body. You definitely want to star mark the specific molecular engines driving this transport. So for forward or anterograde transport going from the cell body to the synapse, the engine protein is kinesin. For reverse or retrograde transport heading back to the cell body, the engine is dynein. And this retrograde path, it is incredibly important clinically. Why? Because this is the exact microtubule highway that dangerous pathogens like the rabies virus and the tetanus toxin, tetanospasmin, use to invade our central nervous system.
डाइनिन इनको literally reverse direction में brain की तरफ ले जाता है.
So we know dynein is essential for movement, right? But what happens if the dynein arm is just missing entirely? Well, we get Carter-Jenner syndrome, which is also known as immotile cilia syndrome. Normally, functional cilia have this classic 9+2 microtubule arrangement, and the dynein arm is what actually forces them to beat and move. So no dynein, no movement. The cilia just strictly halt. Because those cilia simply cannot move, patients present with a very classic clinical triad. Number 1 is bronchiectasis. This happens due to severe sputum accumulation in the lungs because the respiratory cilia just can't sweep it clear. Number 2 is infertility, which is due to absent motility in the sperm and the fallopian tubes. And number 3 is situs inversus, where the internal organs are literally rotated onto the wrong side of the body because ciliary movement is strictly required for proper organ rotation during early embryology. Also, please don't forget the pharmacology connection here. Microtubules are directly targeted by anti-cancer drugs like vinca alkaloids and taxanes to stop cell division, and by colchicine for gout. Ye pharmacology ka bhi high-yield point at, and interlinking these subjects is the real key for NEET PG. Okay, next up is Section 3, microfilaments and listeria hijacking.
We know microfilaments mostly consist of the proteins actin and myosin. And we all know their vital role in skeletal muscle contraction through the sliding filament theory. But prepare yourselves for a really fascinating microbiology crossover regarding cell motility. Listeria is this incredibly clever bacteria that literally hijacks our own host cellular machinery to move around. It doesn't just swim on its own, it physically forces our actin microfilaments to push it forward. Let's walk through this pathogenesis step by step, just like we're mapping it out on a whiteboard. First, listeria uses internalins A and B to force its way into the host cell, where it immediately gets engulfed into a vacuole that tries to contain its spread. But to escape this trap, it secretes listeria lysine O, which completely breaks down that vacuole. Now here is where the hijacking happens. The bacteria uses a specific protein called Act-A to stimulate massive host actin polymerization right behind it. This rapid, intense buildup creates a classic comet tail pattern of polymerized actin that acts exactly like a rocket engine. It propels the bacteria forward, creating what we call tumbling motility to shoot into and infect nearby healthy cells. Concept ekdam clear hona chahiye yahan. Let's hit section 4, intermediate filaments as tumor markers.
Students, listen very carefully to this part because this is an absolute goldmine for pathology MCQs. Intermediate filaments are highly cell-specific, which makes them the absolute perfect diagnostic markers for identifying exactly where a mysterious tumor originated. You really want to take a mental screenshot of this categorization. You have to match the protein strictly to its marker. For example, keratin is for epithelial cells, commonly seen as a marker in squamous cell carcinoma. Desmin is found in muscles, so it marks sarcomas like rhabdomyosarcoma. Vimentin points straight to connective tissues, meaning tumors of mesenchymal origin. GFAP is for astrocytes, so it marks central nervous system gliomas. And lamin, which is located inside the nucleus, is famously mutated in progeria, that premature aging disorder also known as Werner syndrome. ये table आपको direct exam में question solve करवाएगा.
And hey, speaking of keratin, in pathology, you are going to hear the term Mallory-Denk bodies over and over again. But what actually are they on a structural level? They are quite literally just pathological clumps, massive depositions of keratin, which, as we just learned, is an intermediate filament sitting inside a damaged liver. This is classically seen in alcoholic liver disease. Reta nahi marna hai, just logically remember the equation. Liver damage plus clumped keratin equals Mallory-Denk bodies. Moving on to section 5, cell-cell adhesion junctions. So we spent a lot of time on the internal skeleton. Now let's talk about how these cells physically hold hands and tightly connect to one another in tissues. Every major adhesive junction basically relies on 3 main components. You've got a primary protein that spans the physical gap outside between the 2 cells, a linker protein that connects it just inside the cell membrane, and finally, a massive cytoskeletal filament that anchors the entire structure deep within the cell for real structural strength. Let's compare the 2 big adhesion junctions side by side, zonula adherents and desmosomes. For zonula adherents, the primary bridging protein is E-coherin, which links via beta-catenin all the way down to actin. Yade, listeria, we just talked about it. Well, listeria's internalins specifically recognize and target this exact E-coherin to invade our cells. On the flip side, desmosomes use desmoglein and desmocolin as their primary proteins, linked via desmoplakin to tonofilaments, which are very strong intermediate filaments. Because of this incredibly robust structure, desmosomes are super strong and highly abundant in tissues facing high mechanical stress, like your heart, your skin, and the uterine cervix. Understanding the normal structure perfectly explains what happens when things go wrong pathologically. If a patient's immune system starts creating autoantibodies that directly attack and destroy desmoglein, which is that key primary protein of the desmosome, the skin cells entirely lose their physical grip on each other. The catastrophic result of this? Pemphigus disorders. The skin literally blisters and falls apart because its structural anchors have just been chemically destroyed. And finally, section 6, tight and gap junctions. We just looked at standard adhesion, but now we're looking at special junctions that either completely seal off spaces or allow incredibly rapid, instantaneous communication. First up are tight junctions, anatomically known as zona occludens. These are built out of proteins like occludin and claudin. You'll find them exactly where you need a strict, impermeable barrier. Think blood-brain barrier, the gastrointestinal tract, and the kidneys. They strictly regulate paracellular movement, meaning they control exactly what is allowed to slip sideways between the cells. A top-tier clinical fact to remember here: a specific mutation in claudin 16 in the kidneys prevents proper ion handling. This directly causes familial hypomagnesemia, hypercalciuria, which ultimately leads to severe renal stones. Do not forget this one. Next up, we have gap junctions. These are essentially tiny microscopic tunnels that allow for rapid communication through a 3 to 4 nanometer gap, and they use integral proteins called connexins. A mutation specifically in connexin 32 directly leads to Charcot-Marie tooth disease. This is a very well-known peripheral sensory neuropathy. Notice how we are directly linking a tiny microscopic gap junction defect straight to a major clinical neurological presentation. To quickly wrap up our summary on gap junctions, always remember they are highly abundant in cardiac muscle and nerve fibers, simply because those tissues need to fire together rapidly and simultaneously. In fact, a mutation in connexin 40 affects the heart, directly leading to idiopathic atrial fibrillation. And finally, don't forget the junctions connecting the cell to the basal lamina. Hemidesmosomes attached to intermediate filaments and focal adhesions attached to actin. Why does this basic anatomy matter clinically?
Well, for cancer cells to become metastatic and spread throughout the body, they must first disrupt these exact foundational cell-to-basal lamino anchors so they can break free.
And that brings us to a closing, provocative thought for today's explainer. If a single structural mutation, like missing just one tiny dining arm or having a flawed desmoglein protein, can cause your internal organs to be completely reversed or your skin to literally blister off, just how fragile and yet incredibly elegant is our cellular scaffolding? Keep that in the back of your mind as you study. These aren't just dry textbook facts to memorize, they are the literal physical building blocks of human health and disease. Keep revising Concepts Clear Reco, and you'll absolutely crush those exams. Until next time, stay focused and happy.